So I want to say thank you all for coming, and pardon my complete, absolute lack of German. So thank you for bearing with me today, and also a deep thanks to the organizers. It's really an honor to be here to talk to you today, really about how to look forward. So I think we all know why we have to get there, and now we have to think about how we're going to get there. I want to briefly go over some of the grounding principles of why we need to think in different ways. When we say we need a paradigm shift in medical research, what does that mean? How do you get there? And you have to understand what components build up that foundation. We have to briefly talk about the failures in therapies for neurological conditions, if only to remember the millions of people out there that are depending on us to make this change. And then I'll go into 3 case studies. Again, these are only 3 examples of how we are innovating, how we are developing human-based models. But what we need, as we've heard earlier in the morning, is that we need hundreds of these approaches, because there are so many aspects in mechanism, in phenomena of disease that we need to understand to develop proper treatments and also prevention. So strategies that not necessarily have to be pharmacological, but have to truly understand and target the mechanisms. And then finally, I'm going to briefly talk about the research pitfalls, also how we are also to avoid being creatures of habit. So not just thinking that we have a rational design, but how something that we make human-based, how can that suddenly fall back into an animal model space? And then finally, rethinking neuroscience from the ground up, understanding sort of the human component to all of this, that when we try to change something as large as research practice, that you have to, it's true, biology will deal with the old guys, but we have to make sure that the tools are there and the principles and the concepts are there for the younger generation to be able to continue to innovate and to continue and to make those breakthrough discoveries that we so desperately need. So very briefly, for those of you that aren't familiar with the Physicians Committee, we are a nationwide nonprofit organization in the USA with more than 12,000 physician members and over 175,000 members. That includes scientists, industry, nutrition, clinicians, all around very much interested in moving to replace animal research, to find human relevant approaches, because that's how we're going to get there with our therapies. We promote prevention, preventative medicine, and also health through good nutrition, and also address controversies in modern medicine, including ethical and scientific issues in education and research. So I'm going to show you a picture of our team in the Research and Regulatory Affairs Department. This is a very rare picture. This picture actually poorly reflects how we spend most of the 365 days a year. But here on this very nice day, we all decide to go kayaking. But what we do on a day-to-day basis is that we constantly work at multiple levels. Here, we just gave a congressional briefing on the Hill a month ago to talk about how do we start to deal with Alzheimer's disease. And we bring in scientists, patient advocacy organizations, because we have supply chain issue questions, we have issues of economics, we have scientific technical issues that we have to deal with, and it's going to take that multi-scale and dynamic network of people to answer these questions. Here is another example of the Physicians Committee where we're promoting the human tissue donation, because again, understanding not just the tools that we need, but also the sources of what builds those human-based systems for research is necessary as well. On the other side, on the scientific research side, primary research is the Green Neuroscience Laboratory, which that I have with my co-director, Ilan O'Hayan, in San Diego. And this is a bit of a broader design, though despite only a much smaller team, a scientific team, where we really think as the animal experimentation and ending that as being part of a larger whole. And that includes how science is impacting the environment, how our ways and our tools and our approaches in science actually change the way we understand the diversity and the necessary diversity of our brain and our behavior, and how we may narrow that with how we go about that with our structures of medicine. We also have to think about issues like autonomy and how, again, as we develop human-based approaches, how that impacts our privacy as we continue to collect these very important human data sets, how that information is being used, how do we protect the autonomy of the individual, so there's an aspect of choice. And there are many other principles, so if you are interested in something to probably put you to sleep with a nice glass of wine, feel free to go to our website and you can read more about our principles there. So let's go to some of those guiding concepts. So what I really want to impress on you today is this idea of multi-scale. So when you start as a scientist, you say, okay, what am I going to look at? Okay, I'm going to look at Alzheimer's disease, or I'm going to look at epilepsy. And then what tool am I going to use? Maybe I'll use an animal, or maybe I'll look at a cell. And then that sort of shows you where you sort of fall in. Are you very low level here in molecules and proteins, or do you go up to the whole, hopefully, human being, and you start looking at the embodied brain, so that's the whole person, or the social context? And then we sort of start getting siloed. We get stuck in these areas. And I think only recently are we not just nodding our heads to say, yes, okay, we need to sort of look at our research and how that references along this whole scale, but how to actually start to work with the data from all these different levels. And the issue, particularly with animal models, is that we get increasingly stuck in these lower levels, and we don't know how to translate up. And that's partially because they're inherently flawed. So we have to understand that interplay. Now, in terms of Alzheimer's disease and related dementia, I think it's no surprise to anyone. If you could, everyone in the audience, raise your hand if you know someone, have a friend or a family member touched by this disease. Just raise up your hand. So take a look around and you can see how large this impact is. And the fact that this was a census done in 2015 by the Alzheimer's Disease International, 46.8 million currently living. This number is supposed to triple if we don't make any progress. And unfortunately, we're getting used to seeing all these headlines month after month, year after year, for decades of failures after failures. And unfortunately, I don't have a pretty slide for epilepsy, but in terms of epilepsy, we have epilepsy affects about 1% of the global population. And of that 1%, 60% of those individuals living with epilepsy do not have any drugs, any way of controlling those seizures. So they have seizures which can greatly affect their quality of life. And so again, same story, decade after decade, nothing really changes. So the question is, what can we do about it? Not why, what can we do about it? So I'm going to talk to you about one of the projects at the PCRM and the Green Neuroscience Lab, which is a Xeno-free stem cell initiative. And this comes from the place of how do we take a human-based approach, such as induced pluripotent stem cells, so taking tissue cells from an individual, and we'll hear some talks right after me that will go probably much more into detail about this, and then reverse engineer them into stem cells so that it can be used in medical science. It has enormous promise in terms of scientific, clinical, and ethical implications. In terms of disease modeling, iPSC with patient-derived stem cells can help us understand functional pathways, so starting to look at those mechanisms. We can look at physiology, so how the actual action happens within the cells of interest, especially in the brain. And we can even, like the first speaker this morning showed, we can, with with organoids, we can actually lay down grids of electrodes, microelectrodes, to start to see how that network activity occurs. So that has great, vast implications for neuroscience. We can look at structure, which is a particular interest of mine because of my background in neuroanatomy, so that we can look at how the function of the brain is being affected by the architecture of the brain. And then we can create novel, better human-relevant models and also improve reproducibility in studies. In terms of regenerative medicine, this term might be thrown around a lot. What that means is that we can hopefully replace tissues that are damaged with tissues that are not. And more importantly, if we can take it from the patients and put it back into the patients, we're going to avoid the risk of xenoinfection. So that means sort of diseases coming from other species. And this is, you'll see the word xeno, that's where this idea of xeno-free is going to come. And then finally, in toxicity testing, there is a lot of great strides. I would say that toxicity testing is ahead of us in terms of medical research of where we are in terms of human-based approaches. But I promise you, I think with enough passion, we will catch up to them because scientists are inherently competitive. So here we go. Here's iPSC. We take it from a patient or a participant, and then we reverse engineer them into these stem cells, which can turn into any cell in the body, and then they can become specialized derived cells, and then we can use them for various things from disease modeling to drug discovery, and then looking at safety issues. Now, what is wrong with this? It's very, very good. But again, when we say that we're not quite there yet, one of the reasons why I'm going to propose is that we have animal-derived products that are contaminating these microenvironments. So remember, these are cells. They need to grow. They need to develop. They need to be changed. And at the same time, even though we're taking the cells from a human, the microenvironments, the food that we are feeding them, are not human. They're not human-derived. And I'm sorry if that sounds a little icky, but let's, if you think for a moment, if you think about how our microenvironments in vivo are working, it needs to be, it comes from us. Our bodies create it. And so, xeno-free simply means no contamination with animal products, and there are various aspects in the microenvironment of the IPS system that needs to change, the media, the matrix, the serum. And the interesting thing is that the regulations for iPSC therapy development for regenerative medicine already demands xeno-free systems. So why isn't every laboratory in the world using iPSCs not having a xeno-free microenvironment? Well, we think it's for three reasons. We think it's an awareness. We actually surveyed and actually interviewed some laboratories, and it was actually quite shocking. These are very good laboratories, but some of them weren't actually sure, or they didn't realize. They gave us a list of their ingredients and said, oh, well, yes, we use xeno-free, and we said, no, this one here actually is an animal-derived product. And they did not know that. So there needs to be a greater awareness. Another is a concept of cost, that xeno-free products are more expensive. And currently, there are a number of xeno-free products that are more expensive, but not by many orders of magnitude. And in some cases, xeno-free products, human-based products, human-derived products, are actually sometimes cheaper. So there's also, finally, the big one, which is research culture. And research culture is a real hard one. And so we continue to hammer with rationality, with evidence, and then hopefully through the scientific method, people will come to see the way. So, however, another reason is that the xeno-free protocols, how you use these products in changing these cells and keeping them happy and then using them in your experience, are not easily accessible, they're not trusted, and they're not widely used. So our solution, therefore, is a xeno-free stem cell toolkit. So this will be, and is being developed, a free online open repository of xeno-free iPSC stem cell protocols. The idea is to create standardized templates with high level of detail so they can be reproduced by any laboratory in the world. If you are a researcher in this audience, you know how hard it is to sometimes communicate with your peers, because the only time we really get to talk sometimes is at conferences. And seeing a person once or twice a year is a very, very poor way of knowing how to do these high-level, detailed protocols and figure out what's going wrong with them. We hope to have an interactive and user-friendly interface with advanced search functions and filtering, and a user experience. So imagine online shopping. You have so much options in terms of reviewing, telling how the product fell apart or which one was better in what situation. And so by having this type of user experience, we will improve reproducibility. We can improve and innovate faster with the improvement to the models, and we will also see evidence with actual method results. So here's the home currently, Xeno-free.org. If you'd like to follow the development of our project, please sign up here. And this is sort of just a snapshot, a snapshot of how the prototype looks like currently. So basically, a user, an end user, or a research lab will submit a protocol, and we put them into a standard template with looking at giving, again, detail on what the reagent and what the setup is. There are images that are available, and then we validate the protocol. So this is where the true challenges come, because what defines a good protocol, right? So currently, most scientific labs deal with sort of a validation of their study is, can I get my stuff published, right? Because once it gets published, then it's industry's work to try to figure out how to make that translate that into medicine, and that's it. So as long as it's peer-reviewed, we're okay. But that's not sufficient. That's not a good indication of whether or not a protocol is good. So we need expert input. We need an advisory board that will review these, and then we need to have particular metrics to determine whether or not, and scores, to see how well that protocol holds up in terms of its detail of information and its feasibility, usability. And then we're developing a process where, again, the idea is not to create barriers for researchers to share their knowledge, but how to increase the quality so that we can eventually, if it's not quite there yet, we work with you as a research laboratory to improve your score so that eventually we can publish it. So that's a Xeno-free. So that's an example of where we come, especially from the PCRM side, where we recognize the huge potential of a human-based approach, and we start to think, okay, well, what's the next step? How are we going to get there? The next example I'm going to give you, which looks at brain metals and synchrotron imaging, is actually more of a preliminary a starting point, where if you're a laboratory and you believe that human-based approaches are necessary, how are you going to deal with some of your research questions? So first, let's talk a little bit about advanced microscopy and imaging. So why is this necessary? Why is this important? Well, to understand disease, we need to know what it looks like. We need to know how it behaves, and we have to know what entry points we can use to change them. So one method is to look, again, from the human-based approach, to look at human tissue. So even human brain tissue, which is sometimes very difficult to come by, or at least at high quality, is exceptionally informative. We can look at the cellular components. We can run assays to look at the protein and gene expression. We can look at the morphology. So the shape of the cells can tell us a lot about what's going on in terms of the health of that cell. And we can look at, like I said, the pathology. And there are many ways that the change in the structure of that tissue can give us information in terms of the connectivity. And connectivity in chronic multifactorial diseases like Alzheimer's and epilepsy is extremely important, because at the level of systems biology, it's that changes in network talk and distribution, which is where I think that a lot of our problems lie. So the quality and accessibility is still lacking. So what kind of method can I develop? This was the question our lab was tackling. What kind of method can we develop that will actually start to address the fact that tissue is hard to come by and the quality is not great sometimes? We still have to do something about that. So, understanding that we have to have methods that have better defined histocharacterization, which is I'm sorry for the scientific jargon, cellular, what the cell looks like. And then issues of low resolution. So earlier we talked about how there are these great in vivo techniques in humans of imaging, like MRI and DTI. But really, those are, you're looking at resolutions of like a centimeter or a centimeter. And what you really need is the micron. So to put that into perspective for those that are not scientists here, that's like saying, you know, your issue, some of the issues that you're trying to tackle are at the brick level of your house, and all you can see is the house. So what we were trying to say is, how do we look at the brick? But the problem is that a lot of the techniques that we currently use to look at the brick destroy the tissue. So how do I make sure I can look at the brick and not destroy the brick at the same time? So it's a lot of little challenges that we're dealing with here. So we thought, okay, we'll start with the issue of biometals. Biometals in the body, and particularly in the brain, are very abundant. So these chemicals, all that periodic table that you learned back in chemistry in high school, those are the building blocks of all the proteins in your body. And that will affect the function of how the cells in your brain communicate through synaptic communication. I'm not going to go through all of these details. I realize that but you know that the cells, how they talk to each other, how the things internally within the cells move around, the metabolism of the cell, so how the cell feeds itself, and how those change. And as you have disease, those things go awry. And then we can also look at larger networks, how the thousands of cells are talking to each other, and how when, for example, something like multiple sclerosis occurs, and that the insulation material starts to degrade all that communication, that highway of information starts to no longer function properly. That is also intimately connected to biometals and how they change. So what we did, and again, I want to say that this is a very interesting story to me, because it goes with something that is really, I would say, very impractical, because you need one of these to do the technique that I'm about to show you. And what this is a particle accelerator. And I'm pretty sure that most of us don't have it parked behind our university. Luckily, in my particular case, I had access to two of them. The one that we used primarily was the one at the Stanford Synchrotron. And so by using these high-energy X-rays, we were able to bombard human brain tissue to look at the brain metals. So this is very exciting. This was another example of innovation where you can repurpose something that was used for material sciences and use it for biological sciences. So constantly thinking outside of the box is absolutely essential if we're going to find new methods of human-based approaches. So we bombard the tissue with high-energy X-rays, and then we look at the resolution and the mapping at large scales. This is a section of cerebellum, and we can start to go into finer detail to look at the metal distribution. In this particular case, it's looking at the zinc, the amount of zinc in the brain. And then using, pardon me, we're using another machine at the synchrotron, we can look even finer. So we're really looking at the brick. And actually, in this case, we were probably at this point starting to look at the dirt in the brick. So it was really quite exciting. And so you can start to see things within cells. And what was beautiful about this technique is that we could look at many metals at the same time, and it wouldn't destroy the tissue, so that we could then, once we had done our scans, we could then go back to the laboratory and do all that fine staining to look at the structure and the cellular morphology, how those cells looked. And so that became very, very useful. And what we found was that even we could apply that was postmortem tissue, so that was generously donated by a patient after they died. Or you can even work with resected tissue. So this is surgical discard from epilepsy patients. So epilepsy patients, one of the ways they can, if they're part of that 60% of individuals that don't have drugs that can stop their seizures, one method is to have epilepsy surgery, where certain areas of the brain can be identified, and if it doesn't affect too many other functions in the brain, can be removed, and that can often lessen, or even in some cases, thankfully, stop those seizures. So what we could do then is put row after row, or column after column, excuse me, of patient tissue and look at how the lesions in the brain were different based on their metal composition. So this really wasn't seen before, and so this was very informative to us. And so what we thought was that maybe this could be used to compare differences in various human-derived systems. So in the right corner, we have an example of actually IPSs derived from control, and looking at how that compared to the tissue that was being extracted. So hopefully this could become a validation tool and pair with other complementary histochemical techniques. So the final case I want to talk about is computer modeling. And so this is really developed mostly for, at the time in our laboratory for epilepsy, but it is starting to connect more and more into the work we do with Alzheimer's research. And so with computational applications, you will hear a lot these days about big data, about deep learning, machine learning, AI, and it has enormous promise in terms of what it can do with these huge data sets that we're collecting on humans through human studies. And so I just wanted to give a scan. Here we have, with clinical trials, epidemiological data, neurogenetics, biomarker studies from those clinical trials. We can start to shunt all of that information. And also here we have all that anatomical work that I was talking about and all that in vivo imaging that I was talking about. You can shunt that all into computer models. And they sort of fall into two categories. You either create detailed models, which are very, very data intensive, and we often call them biological realistic models. So they're supposed to sort of look like the human system they're looking at. Or we can create abstract models. And what I'm going to focus on is the promise of abstract models today. So this is the work done by my co-director, Ilan O'Hain. And what he does is that he creates these models of the brain structure, these geometric models of the brain, and let these simulations see how that activity of the brain works, or these networks work. And then by looking at them, he can tune them to see how they change over time. And then again, these are all computer simulations. But what's interesting is that then you can compare them to, sorry, I'm having a bit difficulty with this pointer. You can compare them to human data, which is up here. And then you can start to see how they look the same and how they look different. And what's interesting about computer modeling is not necessarily about how much we can make the computer model look like the biological data that we're getting, but trying to explore the spaces that we're not seeing with the human data, because therein lies where we might find how to get to a healthy system. Ilan often says to me, it's very easy to make computer models sick. I can make most computer models look like they're going into epilepsy. What is hard is keeping those systems healthy. And that is where we generate those new hypotheses so that we can start to think, what are the mechanisms that keep the brain healthy? So here, his model runs, and then we see inhibition, the cells that inhibit certain aspects of brain activity, and the other that propagate and spread through the brain. And then we can plot the activity. And again, what we need to do is know that there are, if anyone says we don't have enough human data to do human-based studies, just Tell them you saw a slide at this great conference and that there's actually quite a lot out there. We have detailed information with very good temporal resolution, so things that happen very fast. We have information of how the cells look like. We know what pathology can look like in humans. We know how they look in very long connections. We can see how they look if you're talking about them in metals. And you can even do finer recordings that are invasive through willing, consenting surgical patients. Here's an example also just to tell you that, again, if you plot some of these techniques and look at what kind of information they give you, you can see we do a very good job with human-based approaches to cover the span of functions that we need to start to develop these hypotheses. So, in my last minute, I'm just going to talk about avoiding the pitfalls of developing technologies. And what do I mean by that? I say they get good ideas that get repurposed back to the habit of animal models. And what I mean by that is the things that are upcoming, the idea of creating human, non-human animal chimeras, so putting human cells into animals to create this hybrid, growing human cells and organs in animals, computational models that are highly dependent on animal experimentation data, which means that if you're getting flawed data in, you're probably going to get flawed data out. And also clinical neuroimaging that is applied that should be good for humans, but then for some reason they think, okay, well, let me just put it back into my mouse. So how do we avoid that? Well, 3 ways. Here's the intersectionality. And this is what we do a lot at PCRM. It's research, it's policy, and training. First, we need to provide regulatory guidance that require studies to have human-based evidence of safety and efficacy. 2, we need to make sure that we can empower researchers by having grant prerequisites that detail the potential of phenomenological, so the phenomena of the disease, the mechanism, and therapeutic insight, and not just in this sort of way. Details. 3, we have to launch research initiatives to study human-only data sets in predictive value of safety and efficacy, because if we don't have that, if we can't have that justification, we're not going to be able to shift things forward. And then 4th, we often don't talk about this, but it's a very important conclusion. It does speak to many researchers. We have declarations of ethical imperatives. So we don't use animals because they're different from us. Absolutely true. But in the way that they are like us, it is unethical. And this is something that we can say plainly and also pair that with the research evidence. We have to improve research to therapy pipeline with evidence-based information modification. So this will know that with the promise of reducing the number of studies and increasing the power of those individual studies will actually make research development therapy much cheaper. And we have to increase this multi-scale research. So understanding that if we keep looking only at the bricks of the house, you're not going to know how to build that city very well, because it takes different types of information and knowledge to do that. And then finally, we have to support next generation human-based researchers to explore basic science and medical research questions. Okay, I lied. A few more slides. Education. I have to talk about education. And the reason why is that we need fundamental changes in neuroscience to integrate ethics and scientific relevance. We have to know that universities are teaching human relevant methods so that researchers know, students know that they have the option of going into these fields. So we've done, at the Green Neuroscience Lab, we're starting to do evaluations of curriculum. We've done studies of looking at surveys of different universities and what they're offering in terms of both ethics and human relevant models. And sadly, currently, not much there. However, we did present this at the Society for Neuroscience 2 years ago and raised a lot of eyebrows and a few frowns. And like I said, scientists are competitive. So next week when I go back to them, I'm hoping that maybe someone will have been spurned to do something about this lack of training. And so finally, we're also doing studies to understand what the cultural barriers, scientific cultural barriers are with scientists to making these shifts. And then hopefully all together, we're going to make a huge difference. So in conclusion, integrative ethics and advancing human-based approaches can help overcome failures in translation in medicine, and developing new human-relevant insight into the brain. So having foundational knowledge that is coming from a place that makes sense. We need to develop critical mass in open science projects that may initially be complicated to organize, but will, in the long run, save us a lot of human capital in terms of dealing with the limited scientific resources that we have. And then finally, education and training are the keystones in moving forward to replacing animal experimentation with effective human-based approaches. And even laboratories, for those skeptics that may still be out there in the laboratory, I think we can all agree that with subscribing to these approaches, you can still appreciate rational research design and knowing that if something is broken, that you do fix it, and that will bring needed insight for personalized medicine in the future. And that is where we're going and can only go with human-based research. Thank you very much.